Good afternoon, everyone. My name is Hedieh Torabifard, and I'm a new assistant professor in Chemistry and Biochemistry department in NSM. I received my PhD at Wayne State University in Detroit, Michigan, and then I did 3 years of postdoc at University of Michigan before joining UTD. I'm very excited to be here, and I'm looking forward to collaborating with both experimental and computational groups. My research is very interdisciplinary, and it is at the interface of chemistry, physics, computer science, biology, environmental studies. So there are a lot of opportunities to collaborate with different groups. So let me tell you about the primary goal of my research lab. We would like to study complicated and unsolved biological problems using computational and theoretical methods. Currently, we have two main projects. For the first one, we would like to study membrane transport proteins. These are very important and the malfunction and disorder of these proteins are associated with different diseases. These proteins are the molecular gatekeepers of the cell that basically imports the vital nutrients and export dangerous toxins within the cell. There are very different types of transport proteins, but initially we would like to study anion transport proteins because for so long anion transport proteins and in general anions were neglected in the field. People were more interested in cation and the behavior of cation inside the body, but there are so many diseases associated with the disorder of the anion transport proteins. Among anion transport proteins, we would like to start with fluoride ion exporter proteins. And that is because they have recently discovered, so our knowledge is very negligible here, but they are very important. And I'm going to tell you why they are very important. Fluoride is actually a natural toxic to many organisms, including bacterial species. So that is why we use fluoride in oral and dental hygiene. So if you think we use it in mouthwash, in toothpaste, and that is because they can kill the bacteria. However, very recently, it has been shown that many bacterial species developed a resistance mechanism in order to export these fluoride ions from their cytoplasm and actually resist the toxicity of fluoride and actually survive. So this is a survival mechanism that microbes developed. So we would like to understand and use computational methods to gain a comprehensive knowledge about their function, mechanism, dynamics, structure, and in order to develop new antibiotics. These are actually introduced as novel targets for antibiotics. However, these antibiotics are going to be bacteriostatic, meaning that they are more inhibitors rather than killing directly the bacteria. So this study has a major impact on oral and dental hygiene. Besides that, it also affects the whole microbe inside the body because we ingest fluoride. And also, it actually addresses the emerging global crisis of antibiotic resistance. So if you are interested in this type of work and you have expertise in actually making these new designed antibiotics, I would be very happy to collaborate with your groups. Bioengineers and biomedical engineers would be very beneficial for this research. For the second project also, I am interested in designing new ionic liquids using bio resources such as amino acids in proteins, vegetables in fatty acids in vegetables, sugar, and so on. These new ionic liquids are actually developed to overcome the drawbacks of currently used ionic liquids in industry. The currently used ionic liquids are toxic, carcinogenic, low biodegradable, so it is not that green. So in order to improve the green character of these ionic liquids, we would like to synthesize and make these new ionic liquids. So if you are interested, I would be happy to collaborate here also if you are using ionic liquids in your research. Thank you so much.